Okay, so this is a good summary image as we continue on with part 2 of our chapter 26. So this shows you different parts of the nephron and their major functions as a good overall summary. Okay, so here's your renal corpuscle. Remember this part right here, your PCT, your loop of Henle, and then your DCT. And then we have yet to talk about the collecting duct and the papillary duct in detail yet, but we'll talk about those in a bit. Okay, and it tells you what function it mainly is in different parts of the nephron. Okay, so this is a good review image and diagram. So remember in the blood vessel chapter, we talked about the detailed process of capillary exchange where pressure at the arterial end changes down the capillary into the venous end. And that was what drives the filtration and reabsorption of nutrients and wastes into the surrounding tissues and organs. And remember, all that was driven by the pressure that your heart is pushing blood through the arteries. Now, in the same way, the same concept of pressure applies to kidneys. It is also driven by the pressure of blood coming from the heart. This pressure is what drives glomerular filtration. Okay, same concept as your capillary system. Now, we just talked about the components of the filtration membrane. Okay, it serves as the main purpose of stopping large solutes like proteins and red blood cells from entering your urine, but allow small solutes like ions to pass through into the filtrate. Remember the word filtrate, okay? Filtrate means those that are going into your urine, okay? So don't, sometimes students get confused with the word filtrate and what's going back into your blood, okay? Filtrate means those that are going into your urine and excreted out of your body, okay? So again, the filtration membrane is made up of your fenestrated epithelium right here, okay? Fenestrated epithelium, which is this red capillary endothelial cells layer that will prevent passage of red blood cell from within your capillaries into the capsular space, okay? Because what this ultimately is, it is a cross section of this glomerulus layer, okay? So whatever comes out of the podocytes is what will enter into the capsular space and then goes into the renal tubule to form your urine, okay? So the fenestrated epithelium are those exhibit by your red blood your blood vessel going into the glomerulus, and then you have your thin layer of basement membrane that connects the capillary cells to your podocytes. Okay, basement membrane will allows some small plasma proteins, nutrients, and ions to cross into it. And then these foot processes, you see these spiky things that you're seeing around, those are the foot processes of those podocytes that are wrapping around your capillaries, okay? Those will be what prevents most small plasma protein from even going through. So here's those pores from the fenestrated capillaries, allows those things to travel into the basement membrane. And these are the foot processes of those podocytes, which will then only allow water and small solutes to go through. So even though some plasma protein may try to escape out of the basement membrane, but it can't cross between those holes of your podocyte. So that's why these three layers of the filtration membrane is what ultimately able to achieve filtration, okay? Where big things go back into your blood, out your efferent arteriole, small things go into the capsular space, into the renal tubule for further reabsorption and secretion, okay? So at the end of the filtration process within your glomerulus, you'll end up with just dissolved ions and small organic molecules that will end up in your filtrate, which will become your urine. That is why the filtration membrane of the glomerulus is acting like a filter in drip coffee, if you like to think of it that way. You know how there's a little paper filter where you put coffee grounds on top, and then liquid that comes out on the bottom will be collected in a beaker, or cup. So that filter is like your glomerulus, okay? And then those grains that's preventing from going down into your cup are like the plasma proteins and red blood cells in your blood. And then you have the filtrate, which is your coffee, okay, coming out into your cup. So that's your urine, okay? Only blocking the big stuff and allow the small stuff to go through, okay? All right. So we are now we are ready to talk about what happens on all those pressure within the glomerulus. Like I said, this is very similar to your capillary system. Okay, so within the glomerulus, there are again some hydrostatic pressures to know, just like your capillary exchange in the blood vessel chapter. For the glomerulus hydrostatic pressure, it is actually slightly higher than your capillary hydrostatic pressure, okay? It is actually 50 millicurie instead of 35, okay? And I'll explain why that is in a bit, okay? For peripheral capillary, we said that CHP, okay, capillary hydrostatic pressure was 35 millicurie, which means for here in this case, your peripheral capillaries is 35 millicurie, but those GHP, glomerular hydrostatic pressure, it's a little higher because there's efferent glomerular arterioles versus afferent arteriole. Efferent arteriole, which is this one right here, it's smaller in diameter. Okay, smaller in diameter than your afferent arteriole. So this is also another way you can identify afferent versus efferent on model in lab exercise, because the thicker one, okay, is your afferent arteriole. The thinner one is your efferent arteriole. Because it's smaller here, it takes more pressure for a blood to be able to push through all these capillaries and out the other side of the efferent arteriole. So that's why you need a slightly higher pressure in order to be able to push blood through. So that's why it's 50 and not 35 for glomerular hydrostatic pressure. If your glomerular hydrostatic pressure gets lower than 50, then no filtration can happen within the glomerulus. Okay, we'll talk about this in a bit. Now, remember that because we did talk about in the blood vessel chapter that the smaller the diameter, the higher pressure you need to push through. So that's why vasoconstriction can lead to a higher or increase in blood pressure, whereas vasodilation can lead to lower blood pressure. So same idea, okay? This is a good thing to remember when you're differentiating between afferent and efferent, arterioles on the lab models, okay? All right, now a second pressure is called capsular hydrostatic pressure. This is talking about how much resistance there is that opposes your glomerular hydrostatic pressure due to that fluid being in your renal tubules and within your capsular space. So that's why you have 50 millicurie of pressure going this way, because that's the glomerular hydrostatic pressure coming through. But you have liquid here too, all over the place here. So that liquid is going to exert a force backwards, okay, which is about 15 millicurie due to that resistance, okay? And now, hence, the net hydrostatic pressure, or NHP, is the difference between the two. One's that's going this way, pushing fluid through this way to form your filtrate, and then your fluid that's actually pushing backwards on the glomerulus. So hence the NHP, net hydrostatic pressure, is 35 millicurie, okay? And we're relying on this pressure to push water and solutes out of the bloodstream into your glomerulus, okay? Now, remember when we talked about filtration in capillary, we had to calculate the net filtration pressure, which was NFP. So NFP is the same formula as we have seen, but now instead of your CHP, we have your NHP within the glomerulus. Okay, so to calculate this NHP was this formula right here, which accounts for what's pushing outwards into your renal tubule, and then your capsular hydrostatic pressure that's accounting for pressure that's going this way. Okay, so that's why your net hydrostatic pressure is this one right here that we calculated, which is 35 minus BCOP, just like our usual NFP formula, which is 25, as we have calculated. We have said that average value is, and hence you have about 10 millicurie of pressure total going this way due to all that pressure existence, okay? And that's why you have this 25 millicurie going this way as well due to the presence of stuff within your blood, okay? So 10 millicurie is that average pressure that's forcing water and solutes from your glomerulus into your renal tubule or capsular space to form your urine, okay? The glomerular filtration rate is how much urine or filtrate your kidney is able to reduce each minute. And this value depends heavily on your NFP value, okay? For example, if your blood pressure decreases just by 20%, okay, which means that glomerular hydrostatic pressure in the beginning will also decrease by 20%. So 20% of 50 goes down to 40, okay? That 20% decrease. You think, oh, that's not much, only 10 millicurie difference. Well, if we continue with our formula to calculate whether any filtration happens, remember NHP is your GHP minus your capsular hydrostatic pressure, CSHP, which then you get 25 millicurie as your net hydrostatic pressure. NFP is NHP minus BCOP. 25 minus 25, 0, okay? When you have 0 millicurie going through your glomerulus, your kidney filtration completely stops, okay? So that's why blood pressure dropping just by 20% is going to stop your kidney from doing what it's supposed to be, okay? So that's why I said it is very critical for your kidney to be able to adjust the pressure that's going in to your glomerulus, or else your entire kidney filtration system fails, okay? So that's why it is very critical. We'll talk about the different processes your kidney goes through to make sure your pressure within the glomerulus and your kidney overall is always maintained at homeostasis, despite the fact that your systemic blood pressure may be a little different. Okay? All right. So just like previously when we talk about blood pressure, any hemorrhaging, shock, things like that, they will actually lead to decrease in GFR and even acute renal failure because you're bleeding. Okay, lots of things are leaking out of your body. Blood volume goes down, blood pressure goes down, including those that's in your glomerular hydrostatic pressure. Dehydration, same thing. Okay, all of those will lead to decrease in GFR. Less GFR would then mean no kidney filtration, and that would lead to what's called acute renal failure. Okay, acute because these are short-term things that are happening that's causing your kidney to fail, not because of chronic conditions. Okay. Hence, because our kidneys are so vital to cleansing our blood, as I've talked about, it is very critical to maintain the homeostasis of our GFR. Once your kidney stops filtering, we're going to start building toxins within our blood, which then, like I said, the person can die within a few days, okay, if it's not fixed. So again, with the homeostasis of our GFR, it can be regulated again by three ways, as we've talked about: autoregulation locally, hormonal regulation, and neuroregulation in the autonomic nervous system, okay? So for auto-regulation, how we can manipulate GFR, which in a sense is manipulating GHP, your glomerular hydrostatic pressure, is by changing the diameter of these capillaries, okay? Whether it is your afferent or efferent arterioles and glomerular capillaries, those can undergo vasoconstriction or vasodilation to adjust the blood pressure within your kidneys. For example, if your blood pressure decreases systematically, okay, we're talking about systematic blood pressure decreasing, either due to, hemorrhaging, bleeding somewhere, or dehydration of some sort, that's locally, your kidney then can sense that drop in blood pressure, okay, which then causes these intraglomerular mesangial cells, which is right here, all these brown tan area. Those cells actually have actin-like filaments that will contract, okay? And when these contract, it actually wraps around this whole thing, okay? When it contracts, it will squeeze down on the capillaries, okay? Squeeze down on the capillaries, which decreases the diameter of the capillary itself to be smaller. So now it's in this tiny little hole instead of big hole, which then will maintain glomerular capillary blood flow, okay? When you undergo vasoconstriction, okay, you will have an increase in blood pressure. And specifically, this is just locally happening within your kidney. So even though systematically we're having low blood pressure, we're not changing whether your kidney is doing filtration because you have these intraglomerular mesangial cells that's going to do that auto-regulation for you and make sure we have high enough pressure within the capillaries to continue doing filtration, okay? Now, if systematic BP or blood pressure increases, this will stretch the walls of your afferent glomerular arterioles, okay, which then will cause smooth muscles to contract and reduce the diameter of your afferent arterioles to decrease the glomerular blood flow. So if we're talking about here's your glomerulus, okay, here's your afferent, here's your efferent. It's a little smaller than your afferent, and here's the rest of your capillary, okay? Blood pressure increase. Lots of blood coming through, okay? Lots of blood coming through the arterial wall of the afferent will then stretch this to be bigger, okay? This triggers these smooth muscle cells within the arterioles, the afferent arteriole now to contract, okay? Well, we don't want outside systematic blood pressure to be affecting what's in here, or else it would affect our filtration rate, okay? So to stop that effect from your systematic blood pressure, this will then squeeze us down, okay? So now you have a smaller blood flow, a smaller afferent arteriole, which then will increase your pressure around that area and decrease blood flow. So regardless, you have lots of blood trying to get through here in the afferent arteriole, all that blood is not going to try to rush through your glomerulus because your afferent can squeeze down to allow less amount of blood to flow through into your capillary, okay, of the glomerulus, okay? All right, so I know it's difficult. I know it sounds complicated, so you can pause and replay back until you understand it, okay? Or you can try to look up on YouTube for external resources to show you how that works as well, okay? Now, with hormonal regulation, this is where we mentioned in previous chapter, and I said don't worry about it until the urinary system, so here it is. Hormonal regulation of GFP is monitored by a system called RAS, which stands for renin, angiotensin, aldosterone system, and by your opposing hormone, which is natriuretic peptides, okay? Natriuretic peptides, these two are antagonistic interaction. Remember those hormone interaction I was talking about back in endocrine chapter. So renin, angiotensin, and aldosterone is an antagonistic relationship with natriuretic peptides, okay? And I'll talk about what this means. So the kidney will release the hormone renin, okay, when you need to restrict water and salt loss in the urine. Let's say you don't have enough water content in your body, so we don't want to excrete as much water out. And that's when your kidney will respond by release renin, okay? So now decrease in blood pressure in the glomerulus due to decrease in blood volume, systematic pressure, and blockage in renal artery will all cause the hormone of renin to be released, okay? On the other hand, if blood pressure gets too high, which means there's increase in blood volume, then we would need to increase GFR to help get rid of all that blood volume and increase urine loss, okay? So that's this right here, blood pressure increases. This is when blood pressure decreases. So when blood pressure decreases, what happens? Renin is released from your kidneys, okay? So blood loss results in lower arterial blood pressure. which means lower GFR. Uh-oh, trouble. We don't want kidney to stop functioning. So kidney right away releases renin, which induces or activates angiotensin. It actually goes through angiotensin 1 and angiotensin 2 through the angiotensin converting enzyme. But you don't have to know that too much detail. But as long as you know renin activates your angiotensin, okay? Renin activates angiotensin, which then activates three things in your body. Increase in ADH. Remember, we already talked about what ADH does. It increases water retention, okay? Increase aldosterone, which will then lead to salt retention, okay? Sodium ion retention. And remember, water always follows salt. So if there is more salt in your kidney, there's going to be more water in your kidney as well, okay? Which then goes back to your circulatory system, okay? So that's why ADH and aldosterone always go hand in hand, okay? If there's an increase in ADH, there's got to be an increase in aldosterone because they go together to help with water retention. Now, angiotensin 2 will also increase your wanting or your brain to tell you, hey, you're thirsty. You got to go drink water. Okay, so that's what's going to increase your thirst emotions. And then that will increase water intake to satisfy your body with more water again. Okay, so make sure you know this chart right here that summarizes how RAS system works to increase your blood pressure within the kidney back or also increase your blood pressure systematically as well. So now, when blood volume increases instead, this is when you have too much GFR, okay, which means then it's going to speed up that fluid loss in your urine, okay? You have too much blood volume, so let's try to get rid of that fluid now. We don't want to try to retain fluid. We want to get rid of that fluid, okay? And so natriuretic peptides is what triggers dilation of your afferent glomerular arterioles and constriction of efferent arterioles. So remember, again, going back to your little drawing of the glomerulus, here's your afferent, here's your efferent, okay? And then here's those glomerulus capillaries. So if you make this bigger and yet make this smaller, okay, squeeze this down, make this bigger, okay, lots of blood are then going to be able to come through, causing an increase in GFR. Lots of blood is going through your glomerulus. Lots of fluid is coming out into your capsular space and also your renal tubule. And that will, in a sense, will cause more fluid loss, okay? And that goes straight into your urine and results in decrease in blood pressure because you have decreased blood volume by getting rid of all that extra fluid, okay? So two different things happening depending on whether you have low blood pressure, okay? Or high blood pressure, okay? High blood pressure, low blood pressure, and how hormones are helping you either to retention of water or get rid of that extra water, okay? Both cases would affect your blood volume, in which then brings back your blood pressure back to normal, okay? All right. Okay, so lastly, you have autonomic regulation. You can have sympathetic activation, which means you have vasoconstriction of the afferent glomerular arterioles, which then decreases your GFR and slows the production of filtrate, okay? This can be triggered by acute decrease in blood pressure or heart attack, okay? Remember, sympathetic activation is your fight or flight system. Remember, I said fight or flight. The last thing you want to think about is peeing. So that's why you don't want to pee, okay? And hence you have vasoconstriction, okay? Glomerulus, afferent arteriole, efferent arteriole. You make this smaller, okay? And hence less blood is going through, okay? And that will decrease your GFR, okay? Less blood going through, less things being filtered, less urine going through. And that's why you don't tend to want to pee when you're in sympathetic activation, okay? Now, the other thing that can cause a decrease in GFR is during strenuous exercise under warm weather conditions, okay? So when you're exercising really strenuously, I'm talking about like running a marathon or something, okay, it causes vasodilation because you're hot, okay? When you are hot, this is something we didn't go over from the chapter 25 that I was talking about. But when you're hot, heat can escape out of your body by vasodilation, okay? Well, vasodilation, remember, decreases your blood pressure, which then shunts blood away from your kidneys and decrease GFR, which is not a good thing, okay? So that's why a lot of time marathon runners can end up with metabolic waste buildup because you have that decrease in GFR due to doing all that strenuous exercise in, let's say, 100-degree Texas weather, which is crazy, okay? So a lot of times marathon runners afterwards have to do a lot of things to make sure they get their body back to normal status, because they can have what's called proteinuria because there's damaged glomerular cells due to hypoxia, okay, because now you have not enough blood going to your kidney, which means not enough oxygen to supply your kidney cells, okay? And that can lead to damage of your glomerular cells, which then that's when proteins can start entering into your urine due to those cells dying. And then remember, these podocytes right here is what's protecting big things from going into your blood, into your urine. So that's why then if these cells starts dying, that's when those blood cells can enter into your urine. Or protein can enter your urine. When blood enter your urine, that's called hematuria, because then if proteins can enter, so can red blood cells. Are pretty big, okay? Some plasma proteins are pretty big, okay? So whether it is too much protein, then that starts going into your urine because your podocytes are dying, that's proteinuria. If blood appears in your urine, that's called hematuria, okay? All right, so this wraps up the filtration part of our renal system. You can see why this chapter is very long. Okay? 2 parts together probably will be equivalent to 2 hours or more, and that's why in our normal class, usually my urinary system lecture does go into the lab time, okay? So bear with me because it's a long, complicated chapter for the renal system. Okay, so let's move on to the absorption and secretion, which then happens within the renal tubule section of the nephron. Remember, filtration happens within the glomerulus part of the nephron. Remember, you have your glomerulus part, PCT, loop of Henle, and then DCT, and the collecting duct. So we just got done talking about this part, which is the glomerulus part for filtration. Now we're going to talk about this part, which is the reabsorption and secretion part. Now, within the renal tubule, you have reabsorption of organic nutrients from your filtrate. Remember, filtrate means your urine about to be formed, okay? While some waste material can be secreted if they have missed them during the normal filtration process, okay? All of these movements of water and solutes occur using diffusion, osmosis, channel-mediated diffusion, or carrier-mediated transport. All of these different ways of transport you should have already learned from 1406, and hence this is where they come into play and why it's so important when I went over this in 1406, okay? All right, so let's review the 4 types of carrier-mediated transport, because we all know diffusion, osmosis, and channel-mediated diffusion pretty well, okay? Now, there are 4 types. You have facilitated diffusion, which is either of these 2 types, okay, where the carrier protein transport these molecules across the plasma membrane without using energy, just by following the concentration gradient, either as a channel protein or a carrier protein. Remember, channel protein just have that little hole going through the protein, allowing the triangles to get to this side, or carrier protein, which then the substrate has to bind to a particular active site within the carrier protein, which then dumps the protein to the other side of the cell. And then you have active transport, which means it requires ATP to allow things to travel one side to the other side. Remember, exchange pumps, okay? Remember with the sodium-potassium pump we talked about so much in 2401 and 1406. That's a type of active transport. It doesn't follow concentration gradient, okay? Co-transport means that you have two substrate going in the same direction across the plasma membrane, okay? Whether they need both, usually co-transport means you need both things to be present in order for both of them to be transported to the other side. So sodium chloride, sodium glucose, sodium amino acid, these are all co-transport proteins, okay? Co-transport proteins that will allow co-transport of these two items across the plasma membrane. Now, sometimes you just need to follow concentration gradient of at least just one substrate. Let's say you have very little bit of glucose on one side and a lot of sodium chloride, sodium ions on the other side. It doesn't matter. As long as one of them follows the concentration gradient, you don't need ATP to follow these type of co-transport. Countertransport is when your two molecules are moving in opposite direction with respect to the membrane. Okay, so in here you have sodium and hydrogen ions moving in opposite direction, sodium calcium ion, and then another one we talked about, which was then bicarbonate ions and chloride ion. Remember, chloride shift within the red blood cell. That's another type of countertransport we've already mentioned. All right. So transport maximum is when those carrier proteins get saturated due to presence of too many of those things that need to be moved, because we're relying on all these proteins right here for things to bind to, or either they bind or they don't bind, but they still need to get across these integral proteins. So if you have too much of those stuff, sometimes you can what's called saturate the proteins that's allowing these things to go across. For example, if you flood your system with, let's say again, too much vitamin C that is not being used up in your bloodstream, then they will be saturating the carrier proteins, and hence they will just pass on right by and end up in your urine instead. Okay, this is idea of saturating your carrier proteins in the kidney, called renal threshold. Okay. For example, here you have different types of renal threshold. Our glucose renal threshold is about 180 milligrams per deciliter. If we go above these threshold, then we would have a condition called glycosuria, which means glucose starting to appear in your urine, which is a first sign of diabetes, which is why the myth was that if ants start crawling around your toilet is because they are attracted to those glucose excreted through your urine, okay? And that's not supposed to happen. Glucose is not supposed to go into your urine unless there's too much of it in your blood system, okay? And why you have too much glucose in your cardiovascular blood system is probably because you are not uptaking those glucose into your liver or your muscle cells or as fat cells by insulin. Okay, without insulin, those glucose are going to keep circulating around in your cardiovascular system, and which then ends up going over this renal threshold, which then ends up being glycosuria problem, and which then can tell you an indication that, hey, you might have, onset of diabetes here, okay? Now, the transport maximum values of water-soluble vitamins actually much lower than that of glucose. So any excess vitamins we take from our supplemental vitamins, those multivitamins, or if you decide to take those vitamin C every single day, you will see your urine change in color. Kind of what I said, if you take too much vitamin C and you're not sick and you're not using all those vitamins, it's just going to end up in your urine. As if they, never been useful in your body. And that's why your urine sometimes looks more yellowish or greenish whenever you are taking vitamins, too much of it, okay? But it doesn't harm your body, but it's just coming right out in your urine system, okay? All right. Okay, so now we go into what happens within the different regions of the renal tubule. In the PCT, okay, your proximal convoluted tubule, 60 to 70% of the filtrate actually gets reabsorbed here. Okay, remember, filtrate are those that went into your renal tubules. which is you're about to be formed urine, when fluid will this is where fluid will then diffuse into your peritubular capillaries and back into the bloodstream. So there's these peritubular capillaries that surrounds that PCT area. So all these stuff you're seeing here will then travel to the peritubular fluid, which I was talking about in terms of interstitial fluid. And then it will travel into those peritubular capillary and then bring it back to your venules, back into your cardiovascular system in your bloodstream, okay? So the things that are being reabsorbed here are your organic nutrients, which is your glucose and other organic solutes like amino acids even. And then you have your reabsorption of ions, such as your sodium ions or your bicarbonate ions. Okay, bicarbonate ions right here, okay? And active transport of urea and chloride ions as well all happens by passive diffusion just based on concentration, okay? And then you can also reabsorb a lot of water back into your blood, okay? So you see, now remember reabsorption means it goes back into your blood in your peritubular capillary. Excretion or secretion means they're going into your renal tubule, forming your urine, okay? So what this picture, a lot of students get confused what this picture represents. This picture represents a zoom-in view of the tubule, okay? This is a cross-section of that proximal convoluted tubule, and then it puts you into this 2D view to show you what's going into your urine or filtrate versus what's going back into your blood via your peritubular fluid. So things that are pointing this way is reabsorption, and things that are going outwards is secretion. So this is secretion of hydrogen ions if you have too many of those, too acidic condition, or you can have excretion or secretion of ammonia, creatinine, drugs, toxins, those same waste material we were talking about in the beginning of the chapter that happens at DCT. They don't illustrate it here in the PCT, but it happens within the PCT as well, but they'll illustrate it in the DCT picture, okay? So again, you have glucose reabsorption, sodium ion reabsorption, bicarbonate ions reabsorption, and water reabsorption, okay, within the PCT. Now, the loop of Henle, which is the next section or region of the nephron here, is a main region which will absorb half of the remaining water after PCT. Remember, PCT reabsorbs 60 to 70% of the filtrate, okay? But you have more still in there, okay? We don't want to get rid of that much of our liquid, and hence the leftover liquid will be reabsorbed by your loop of Henle, and 2/3 of the remaining sodium chloride ions are also being reabsorbed at the loop of Henle. These are the only 3 things that are being reabsorbed at the loop of Henle, okay? Sodium chloride and water. Now, what is being moved across the membrane differs within the 3 sections of the loop of Henle. That's why we have to distinguish these 3 subsections of the loop of Henle. Remember, this red area, okay, right here, is the descending thin limb, okay? This section right here is the ascending thin limb, okay? And then you have this section right here is the thick ascending limb. Okay, so let's go through what's permeable, what's not permeable. So within the descending limb, as your filtrate is coming through this way, coming down into the loop of Henle, this membrane of the descending thin limb is not permeable to solutes, but it is permeable to water. And hence, that's why the only thing moving out of the descending thin limb is water. When you get your urine then goes down here, gets to the ascending thin limb, this section here is not permeable to anything, okay? Nothing moves out or in within the ascending thin limb. It is only when it gets to your ascending thick limb then you start to have a lot of reabsorption of sodium and chloride ions, because when you get to the thick ascending limb, they will actively transport sodium ions and chloride ions, but it is impermeable to water. Okay, so remember, the left side of the loop of Henle is permeable to water only, whereas the right side of the loop of Henle is permeable to sodium chloride solutes only, but impermeable to water. Okay, just for now, this is all we're gonna stop at talking about this. We're gonna come back to this idea of countercurrent multiplication when we get to that step, okay? But because this is actually a really cool process that automatically causes your loop of Henle to reabsorb as much water as it can, okay? We'll come back to this idea in a bit. All right, so now the third region is the DCT. DCT, your distal convoluted tubule, right here in this region of the nephron. Okay, this is where you have a few things that are reabsorbed, like your sodium and calcium ions. Sodium ions are reabsorbed via countertransport of potassium. Okay, remember your sodium potassium pump. Okay, same thing here, counter transport of sodium potassium. Now remember from our endocrine chapter, we talked about aldosterone being one of those hormones that is produced to promote reabsorption of sodium ions. It does this by promoting production of ion channels, sodium ion channels, and sodium ion pumps. But if you have abnormal prolonged stimulation of aldosterone, okay, this will end up with too much loss of potassium, because now you have all these extra sodium pumps and sodium ion channels stimulated by aldosterone. Remember, for every sodium that ion that you're reabsorbing, you're secreting potassium, which means if you're reabsorbing too much sodium ions, you're secreting too much potassium ions, okay? So this can actually lead to a condition called hypokalemia, okay, which means you have too little of potassium left in the body, okay? We will revisit all of these conditions again in the fluid electrolyte chapter. For reabsorption of calcium, we already talked about this from the endocrine chapter as well, which involves your parathyroid releasing your parathyroid hormones. If you don't have enough calcium in your blood, then we will just secrete less of it, okay, into your urine so that we can reabsorb all those extra calcium ions back into the blood, okay? So back calcium ions as well. All right, what's being secreted in the DCT, just as we had secretion in the PCT, we have similar secretion of potassium ions as we have already talked about due to countertransport with sodium ions. Hydrogen ions can also be secreted if blood pH is too low or too acidic due to buildup of CO2. Remember, if you have too much CO2, okay, you end up making a lot of carbonic acid, which then splits into hydrogen ions and bicarbonate ions. So increase in hydrogen ions is going to tell your kidney to get rid of those extra hydrogen ions. Okay, so that's why you have excretion of hydrogen ions or bicarbonate, depending whether you're too acidic or too basic, as we have already mentioned. Now remember that your hydrogen ions is actually counter transport with sodium ions. Okay, so another thing that can lead to too much sodium ion reabsorption is if you have too acidic conditions as well. Okay, acidosis. We'll come back and talk about acidosis in the next chapter. Okay. Now, ammonia, which was created by amino acid deamination process. So here's the amino acid deamination, you creating ammonia, which of course can be neutralized by urea cycle, okay? But it can also be gotten rid of directly in the kidney as a toxic chemical, as we've talked about. Ammonia is toxic to the body in chapter 25, and now we can actually countertransport sodium ions with ammonium ions as well to get rid of that toxic stuff, okay? So that's ammonium ion. So this reemphasizes what I was talking about in terms of what happens when our blood pH gets too low or too high. Hydrogen ions will be excreted into the urine when your blood pH is too low, which will acidify your urine, but be able to bring your blood pH back up to normal. And as we learned from the last chapter, blood pH can decrease due to lactic acid acidosis from working out too extensively, or ketoacidosis, which is from starvation or diabetes. Okay, so those are the condition can lead to decrease in blood pH, which then your kidney will be actively doing this excretion of hydrogen ions to maintain your pH level. Remember that aldosterone stimulates reabsorption of sodium ions, and since sodium and hydrogen ions has that countertransport relationship, aldosterone also, in a sense, stimulates secretion of hydrogen ions. Okay? So aldosterone also stimulates hydrogen ion secretion due to retention or reabsorption of sodium ions. Okay, so sodium ions comes in, hydrogen ions go out. Okay, so if your pH is too low, it's going to cause your kidney to increase secretion of hydrogen ions, and then your pH can come back up. If your pH is too high, then it's going to increase bicarbonate ion secretion. Okay, so that's why you have bicarbonate ions here. Depending whether you're too acidic or too basic, it can go either way, okay? All right, so the last portion of the nephron is the collecting system, where you have the collecting duct right here. And then what's being reabsorbed here is any last of the sodium ions that need to be reabsorbed, bicarbonate ions, and urea. Okay, so sodium ions, bicarbonate, and urea. So aldosterone actually stimulates reabsorption of sodium ion all over the nephron. Here is that countertransport of sodium ions with potassium ion again. This process, remember, we talked about earlier, is opposed by your natriuretic peptides, okay? Natriuretic peptides, specifically atrionatriuretic peptides, okay? And then you have bicarbonate ions that can be reabsorbed via chloride shift again, okay? So this is how we are able to have, sort of pH regulation due to either your reabsorbing bicarbonate ions or your reabsorbing hydrogen ions. Now let's come back to what's going on in the loop of Henle. Okay, before that, here's that secretion of hydrogen ions versus bicarbonate. And I think this is the third time I'm talking about it. Okay, we're going to come back to this again when we get to the fluid electrolyte chapter, which includes a section on acid base. Okay, so pH regulation I already talked about 3 times. I'm not going to repeat myself again. Okay, so read through this again if you forgot how your kidney is able to regulate pH. Okay, so let's come back to what's going on about the loop of Henle. Okay, so in the loop of Henle, you have what's called the countercurrent multiplication, where exchange of substances between these two segments of the nephron is in sort of promoted by each other. Okay, you have your ascending thick limb, okay, so your thick ascending limb, and then your descending thin limb here, okay. So what happens is you have reabsorption of water from your descending thin limb. A lot of water going into the peritubular space. So as water is being reabsorbed, as your urine is moving down further into the renal medulla, it gets more and more concentrated. Okay, so this number right here represents the concentration of solutes within your urine. So that's why you go from 300 to 120 down here. Not 120, excuse me, 1,200 is because as that filtrate is moving through your descending thin limb, water is being reabsorbed, okay? So when water is being reabsorbed here, you're concentrating your solutes down by the time you're here, okay? And then you're going to come up here. And now because of that high concentration of solutes, salts are then going to start be reabsorbed within your thick ascending limb. In a sense, this is like a positive feedback loop that as you're increasing your solute concentration going down here, that's going to promote the reabsorption of sodium chloride ions up here, because then you're not permeable to water any longer when you're up here, but permeable to salts, which then you have a lot of, okay? So that's why now you're going from 1,200 concentration of solutes all the way down to 100 when you're done going through the loop of Henle on the ascending side, okay? Now, what drives these sodium ions to move out of the thick ascending limb is water. Remember, water always follows salt. So all this salt coming out is what drives this water coming out on this side, okay? So that's why this is a positive feedback loop. This reabsorption of water is what concentrates salt, allows salt to come out of the thick ascending limb side, and once salt starts coming out into the peritubular space here, or peritubular fluid, this is what's going to tell the water from your descending thin limb to come outwards, okay? So it's sort of what's happening here is promoting what's happening here, and what's happening here is promoting what's happening here. So that's why it's called the positive feedback loop that multiplies the concentration differences between your descending limb and your ascending limb, okay? So this is what's called countercurrent multiplication due to the membrane differences in terms of what they are permeable to. So water is what moves out of the descending limb, while sodium chloride is what moving out of the ascending limb, okay? Now, so all those water reabsorption along the PCT loop of Henle that we were talking about is something that must happen due to osmosis. It is called obligatory water reabsorption because we cannot control this with hormones, okay, even though we want to, but there's nothing we can stop osmosis from happening within PCT and loop of Henle. It's all based on water and solute concentration using osmosis. Whereas facultative water reabsorption is something we can control depending on our fluid intake, okay? We're dehydrated versus overhydrated. This is what we can change, is the facultative reabsorption within the DCT portion or the collecting duct portion, using the hormone ADH. Remember from our endocrine chapter, we talked about how ADH is a hormone that can stimulate water reabsorption. But how does it do it? Now we can understand exactly how ADH can cause water reabsorption. So what happens is when we are in absence of ADH, no ADH, your DCT and collecting system is impermeable to water, which means no water reabsorption. Okay, water doesn't get it reabsorbed. All the water stays within the collecting duct, and hence you end up with dilute volume that looks, sometimes if you drink a lot of water and you go pee and you find yourself, wow, my urine also looks like clear, like water. It's because you're drinking a lot of water and you don't need to reabsorb as much. So that's why without ADH stimulation, you have large volume of dilute urine. Now, with ADH, let's say now you're not drinking water for a couple hours, now you're in dehydration mode, and your body just needs more water, then ADH will be released, an increased amount of it. And ADH is going to cause formation of what's called aquaporin, okay? Aquaporin sounds like water, pores. Okay, so they are literally water channels that can be punched in to the DCT or your collecting duct area. Okay, it's going to have these channels punched in there called aquaporins, and then now water can flow right out through the DCT or through your collecting duct. Okay, so that's why more ADH means more formation of aquaporin, which then allows for more reabsorption of water back into this tubular peritubular space, which then goes into your peritubular capillary. Okay, so technically speaking, your hypothalamus is continuously secreting low levels of ADH, which then makes your DCT and collecting system usually reabsorb about 5% and 9% respectively of water when you have this normal ADH levels. But when you are dehydrating or your body is not having enough water, more ADH will be released by your hypothalamus, and then when you have more water reabsorption, your urine will be smaller volume and it will be more concentrated, and that's when you have more yellow, dark yellow of your urine because you didn't drink enough water. Okay, so ADH, as we've already mentioned, is antagonistic relationship with natriuretic peptides or natriuretic peptides. Okay, so when you have too much water, okay, AMP will be released to release all those extra fluid from your body, causing your blood volume to go down, and hence you don't have, you can control your blood pressure as well. Okay, when you have not enough water, ADH will be released. Okay, and this causes a lot of your urine or water to be reabsorbed, and hence your urine will be more concentrated. Okay, so when AMP is stimulated, you have large volume of dilute urine, which then will decrease your amount of blood fluid. Okay, so let's summarize that a little bit here. So increased ADH is equal to increase water reabsorption, which then equals concentrated urine. Decrease ADH and or increase AMP is going to cause decreased water reabsorption, and you have dilute urine. Okay, so that's why when we talked about earlier, when we talk about ADH, if you drink a lot of alcohol, you are actually inhibiting ADH or increasing the amount of natriuretic peptides, which then that's why after you drink alcohol, you end up peeing more because you don't have enough of those aquaporins for reabsorption of water. Okay, so the next type of exchange we need to talk about is what happens between vasa recta, and which was the capillary that surround our juxtamedullary nephron. Okay, remember we said this is a very special capillary, and it wraps around that long loop of Henle juxtamedullary nephron going into the renal medulla area. It is similar to what was happening within loop of Henle and peritubular fluid with countercurrent multiplication, but what happens in the vasa recta is called countercurrent exchange. So make sure you don't mistake countercurrent multiplication with countercurrent exchange. Make sure you don't mix these two up. So what happens during countercurrent exchange is that as blood descends into the adrenal medulla region through the vasa recta, solute absorption predominates. So all those sodium, potassium, chloride ions is going to go into your capillary because a lot of these ions are coming out of your other side of your loop of Henle. Remember, this ascending thick limb of the loop of Henle only allows ions to pass through, not water. So what's abundant in this peritubular space or fluid here is ions, okay? And hence all those ions are high concentration on this side versus in your blood, so it just flows right into your vasa recta based on concentration gradient differences from high concentration to low concentration, okay? Now, when your blood starts ascending towards the cortex area. Okay, so this, now it goes up into your cortex area. See how your urine moves this way, but your blood moves the other way around, okay? So when your blood gets to this side, trying to ascend into your renal cortex area, remember this side of the loop of Henle is only permeable to water, but not permeable to salts. So what's going to be in this peritubular space here is just water. Okay, and because water is abundant on this side, it's just going to flow right into your vasa recta by osmosis. Okay, so what's happening predominates here is diffusion because you have lots of solutes, and what predominates here on this side of the vasa recta is osmosis, which then a lot of water is going to reabsorb right back into your blood and then up back into your venous system, back to your cardiovascular system. So removal of solutes and water by the vasa recta precisely balances the rate of solute reabsorption and osmosis in the medulla. So you have countercurrent multiplication happening within the loop of Henle that causes your urine to have less water and also reabsorb all those important ions from your urine. And then what about your bloods? And they gotta go in your blood too. So what happens with your blood is through the vasa recta, countercurrent exchange is allowing those salt to go into your blood and those water to go into your blood via this automatic system, just by what's permeable within your ascending limb of loop of Henle or descending limb of loop of Henle is what drives the movement of ions versus water. Okay, so that's for reabsorption of stuff back into your cardiovascular system for countercurrent exchange. Okay, so this is a good overall summary slide for you to look at renal filtration, reabsorption, and secretion in a big picture. This is your glomerulus, so this is where filtration happens, okay? This is your PCT, this is your DCT, this is loop of Henle, okay? And PCT, DCT, you have reabsorption and secretion of stuff, okay? Reabsorption of essential ions like hydrogen ions, sodium, chloride, all those things we talked about, glucose. And then you have loop of Henle, which only involves water and sodium chloride. And then you have the collecting duct, which is where we can do facultative water reabsorption, depending on whether we can have ADH stimulated by making more aquaporins available within the collecting duct to allow more water reabsorption back into our vasa recta or peritubular capillaries. Okay? All right, so make sure you can actually know and understand what's going on in this picture according to what we've learned in all those specific sections of the nephrons. Okay. All right, we are almost done here. Bear with me. Like I said, this is a long chapter with very complicated concepts. So there are few urine exams that can be done to analyze urine composition or how well your kidneys are functioning. In the lab exercise for urinary system, you will do a lab on urinalysis where you can determine the solute contents of your urine, like glucose, proteins, blood cells, which normally should not be in urine. The color appearance of the urine is also a good indication of the health of your urinary system. Okay, creatine clearance is a way we can tell how well our glomerulus is doing the job of filtration. So the composition of urine and concentration of urine is what we're trying to figure out through urine analysis. And these three kidney function tests here, it's what's telling you what's in your urine and what's not supposed to. So if we get to do this lab exercise in real person, you actually analyze your own urine in lab, but since we can't do it anymore, we're gonna do a virtual way through your virtual lab exercise. Okay, so creatinine clearance is an indirect way of telling you how well your glomerular filtration is happening. Okay, if your glomerular filtration rate is happening correctly, you will be able to see that you have less amount of creatinine in your body per certain time. Okay, so that this is, you don't have to memorize these numbers. It's just showing you how creatinine clearance is an indication of how well your glomerulus is doing filtration, filtering those bad stuff from your body, okay, into your urine. Blood urea nitrogen, or the BUN test, is to measure how much urea is in the patient's blood, okay, which shouldn't be high if your kidney is doing its job in removing urea. Urea must be removed from the patient's system. Some of it is reabsorbed, but some of it is removed as well. So it can't, sort of have too high in concentration in the person if your urinary system is doing what it's supposed to be doing. So just as we had defecation reflexes, we also have urinary reflexes. The urinary storage reflex, or known as continence, okay, is regulated by spinal reflexes through your pontine storage center. Okay, your afferent impulses from stretch receptors within the bladder will stimulate sympathetic outflow to your detrusor muscles within the bladder and internal urethral sphincter. Okay, it actually stops your detrusor muscle from contracting, because when you're storing, you don't want your bladder to be doing contraction. So storage reflex mean your detrusor muscles in the bladder stop contracting, inhibit contraction, okay? And then your internal urethral sphincter will contract, okay? Because the sphincter, remember, it's a little hole like this, okay? If it's contracted, okay, then it's going to close up that hole, okay, so it doesn't allow fluid through. When a sphincter relaxes, it opens up that hole, so now fluid can come out of that hole. So that's why when you're trying to store something, you want that sphincter to contract. Okay, so it also inhibits urination by decreasing your parasympathetic activity. Remember I said when you're in that sympathetic stimulation, the last thing you think about is, peeing. So that's why when you're trying to store urine, is decreasing parasympathetic, increasing your sympathetic. Okay. And that's going to increase the motor activity at the external sphincter, okay, which means the external sphincter will contract, okay, which this closes and then prevent urine from coming out of your external urethral sphincter. Now, when you're trying to do urinary voiding reflex is when you go pee, okay, or your pontine micturition center. The afferent impulses from those stretch receptor within the bladder will stimulate your interneurons to relay sensation to your pons that you gotta go pee because your bladder is full now. So that will increase your parasympathetic activity, which is opposite to storage. Your detrusor muscles in your bladder is going to contract. Your internal urethral sphincter will relax, allow fluid to come out, and then decrease sympathetic activity, and also decrease the efferent motor nerve activity at your external urethral sphincter. So both your internal and urethral sphincter relaxes, allow urine to come out, which then allowing you to pee. Now, incontinence is the inability to control your urination voluntarily. Okay, which means you're even though you're trying to stop yourself from peeing, but pee just comes right out. And now this can happen when we have any trauma to your internal or external urethral sphincter, because those ultimately is what's stopping you from urine excretion. Okay, so mothers that have a stress urinary incontinence, if your childbirth overstretches or damages those sphincter, when you're trying to give birth to a baby, that down there is all kind of stretched out to allow the baby to come out. Now, sometimes that can damage the sphincter permanently, and then you would end up having incontinence problems. Or also in older people where there is general loss of muscle tone. Remember, the fact that that your sphincter can contract is due to the existence of those muscles having muscle tone that can contract and close up that hole. But if your muscles is kind of loose around here, then fluid is just going to come right out and leak out. And that can lead to what's called the automatic bladder. Okay, so this is illustrating all that stuff about whether you're trying to store urine or you're trying to pee. Okay, when you're trying to store urine, your detrusor contraction is inhibited, whereas your internal and external urethral sphincter contract. If you're trying to pee, your detrusor muscle contraction needs to happen, and your detrusor muscle contracts, and then those sphincter need to relax in order to allow pee to come out. Okay, and the last thing are the number of diseases that's associated with the kidneys. We will name a few here. You have renal calculi. So renal calculi are another fancy name for kidney stones. Okay, sometimes kidney stones can form within your urinary tract, either in the pelvic area or within the ureter area, due to calcium deposit, magnesium salt, or crystals of uric acid. This can lead to urinary obstruction because then your urine can't go through the ureter into the bladder, and hence this condition is called nephrolithiasis. Okay, nephrolithiasis, urinary obstruction. So usually this causes extreme pain and decreases the filtration because now you have urine backing up into your kidney area, which then pushes a lot of pressure within your nephrons. Okay, which is a bad case. If you have too much pressure coming out of a capillary hydrostatic pressure, then you're going to stop filtration as well. So it affects by increasing your capillary hydrostatic pressure, which then causes your glomerular net hydrostatic pressure to be changed, which affects your filtration rate. Okay, so usually how these renal calculi are getting rid of is by either, hopefully, if your urinary peristalsis can get rid of by itself and you start peeing stones out of your sphincter, then that's how you can dislodge them, hopefully. And that's why they say whenever you have a kidney stone, drink a lot of water, okay? Drink a lot of water to hopefully increase the amount of peristalsis that happens and, in a sense, getting rid of those kidney stones. But if that's not enough, they can be either removed by surgical means or by what's called a lithotripter. Lithotripter is sound waves that's produced by this machine right here, and it's going to emit sound waves into your kidney or area that will break those stones into little fragments. So once they're smaller, you can have an easier time peeing them out. Now, renal failure is when the kidney cannot filter waste from the blood and gradually decrease the production of urine over time. All your entire body systems are affected when your kidney stops functioning. And this is why when your kidney stops doing filtration, you're going to have kidney failure very soon, and the person will die within a matter of days. Okay. Now, renal failure can lead to blood pressure increase, anemia, because now when your kidney is not functioning right, you're not going to have EPO stimulation. So that means you can have anemia, or you have central nervous problems, okay, which are central nervous system problems, which is coma. Okay, so usually people with renal failure, if they can't get a transplant of the kidneys as soon as possible, then they have to rely on what's called hemodialysis, which is a treatment that will cleanse your blood this way by using a magnet kind of thing that will attract all those filtrate or waste into the other side and let your clean blood going back into your system. So dirty blood comes out, go through the pump, gets through this artificial filter that's going to make all that, excrete all those stuff for you, and then clean blood comes back into your body. Okay, so this is how your entire body's fluid can actually be cleansed by something that's external outside of your body and then going back inside. Okay. All right, so this wraps up chapter 26. It's a long chapter, so don't forget that a lot of these concepts will come back in the next chapter on fluid electrolytes. So don't forget this all after exam 3 is over, because we will revisit a lot of these concepts again in the fluid electrolytes chapter when you do your final exam. Okay? All right. See you.